or exercises because you have them already. They are the activities you have done in class. All right? The activities you've done in class are all you need. Like you can practice those over and over again, right? You were given certain genes, you could use the same thing and just switch a gene. Pick one that you're interested in. Right? But there will be no, because it's not like a normal, so think of, like, remember, we're assessing skills. Right? So if all I do is teach you how to do a skill, I'm not going to change the skill when you get on the test, right? It's not, there will be some theory on there, but those are questions you will already know basically from doing the applied stuff in class. So there are no practice questions. Well, again, run through the activities. The more you can do them, the easier it becomes. You'll be fine. Any other questions? Any other questions? No? Reminder, the drop-in session this week is on Thursday morning, I believe, which is tomorrow. The location is, as of right now, the location is the Veterinary Science Conference Center on the other side of campus. However, there was a fire in there yesterday. So it might change, but if it is changed, I will post an announcement, and I will also change it on the calendar on the page. It is in person only tomorrow, I believe. Any other questions about that? All right, so if there's something you're unclear of, or you want a little more individual help or assistance, I would strongly recommend the drop-in sessions if you are able to attend them. All right, so we will pick up. Hopefully everybody's virtual machines are now up and running. I can see many of them are on. We'll pick up where we left off. So how many of you should already have the individual file there? You should be seeing something we see here on the screen. Right? Oh, sorry. Okay, so remember what we've done in class here is an example data set. So what you see here in this .hom file is the individual, you see individuals, but it's also the individual run of homozygosity. So it's not the total runs of homozygosity for that individual. It's just the individual, every run of homozygosity it has, it's going to have a line in that file. So if the very first example, the AUR, had more than one run of homozygosity, then you would see that individual in there twice. You'd see AUR twice, and you would see chromosome 16, and then maybe chromosome 10 or 12, with the listing of the other runs of homozygosity. Right? The other file, the individual one, gives you essentially a summary of that information per individual, but you could find it by the .hom file as well, simply by adding up the KB lengths of those runs of homozygosity to give you the total runs of homozygosity for an individual. Right? So I had a good question at the end of class was, what question might you be asked in real world scenario that you might need to do this? Well, the simple question is, you might be asked, oh, I have a pedigree animal, or I have I was saying it just happened in the last few weeks, hey, we've got this population of horses, we're curious if they're inbred. What is the inbreeding of these individuals? So there is no pedigree on them because they're a wild population, so we put them on the genotyping array for horse, and we run runs of homozygosity analysis, and that gives us the inbreeding coefficient for each of those individuals, and then on average I can say, okay, the average inbreeding coefficient for this herd is 0.25 or something like that, right? Because this gives you the inbreeding coefficient. So hopefully, if you remember from right before the break, when you calculate the inbreeding coefficient for this, the AUR individual at the top, it only has one run of homozygosity, which is 1,034 kbs in length. When you divide that by the total size of the genome, which we said was what, 650 megabases? Megabases. You get a very, very small inbreeding coefficient, pretty negligible, so that individual seems to be quite heterozygous. Not a lot of inbreeding for that one. All right. Any questions about that? I see a hand go up. No? No? All right. So, but how about the other summary file, right? So we said there was two files, the individual one and the HOM summary, right? So that HOM summary file reports the number of individuals that each marker in a run of homozygosity, that have a marker in each run. So the individual file gives you a summary per individual, right? The HOM summary file gives you summaries per SNP, per individual marker. Right? So it separates the individuals according to their phenotype, cases and controls. They're all -9 here, but in week 9 we will start talking about genome-wide association analyses. Right? And this file tells us about population level fixation. So similar to what we mentioned earlier with the Dobermans, population level fixation. So the level of population fixation can be determined by the proportion of all the individuals that are homozygous. If more than 90% of individuals are in a run of homozygosity include that marker, then it could be considered as near fixed, right? So a lot of times what will happen in terms of the applied aspect of this is we might have a population of individuals, and we might say, all right, how much diversity is there? We want to have a breeding program. It could be something that's tied to the zoo. And we might do a run of homozygosity analysis across each individual, right? So if you remember earlier in the slides, you would have seen the picture that said, okay, the things in blue are, light blue were homozygous and dark blue were heterozygous. Everybody remember that, right? Well, what should we be interested in is like, okay, are there any shared homozygous regions across every individual in our population, right? And if we can show that like these single SNPs are homozygous in 90% or more, they're essentially approaching fixation. And this can be something phenotypic, so in standard bred, so like harness racing horses, they're fixed for a certain thing that allows them to perform really well at the racetrack. They're essentially fixed at that location, right? In a dog breed, it could be something as simple as they have sticky uppy ears or floppy ears, right? And if it's something that's really interesting is if it's a breed that has a unique trait, it's probably in that area because it's set to the trait. If it's something that you're unsure of, you can do what? Well, you can use the genome browsers, figure out, okay, this region is homozygous in the vast majority of these individuals. And then you can put that region into one of the genome browsers for your species of interest, and you can see what genes are in there. And you guys should know now, right? Okay, you see the gene, you can also look up what those genes do. You can use OMIA or OMIM to see what those genes have been associated with in other species. So you can find like, oh, okay, this is highly conserved in this wild population of 100 little ponies, right? Sorry, my examples will mostly be horse. And you can look it up and be like, oh, this is actually associated with a certain color, and this population is fixed for that color. So you can use this information in a variety of ways, and it's mostly going to be looking at breed and individual diversity, right? And so from there, the order of the files to the output files are important, right? So the order of the output file for the summary file is chromosome, marker identifier, marker chromosome, position, number of affected individuals that are homozygous, and finally the number of unaffected individuals that are homozygous. It is up to you to know how many individuals are being considered. You should know how many are in your population. We did it earlier before the break. How many individuals do you have in this population? 85, correct. All right? And you can also find what is in the file by looking at the help page, right? Just like before, it'll give you a nice little breakdown and list and detailed explanations of what each column does and how it's done. So having said this, how many lines are in your home summary file? Now, I would strongly encourage you to consider not catting the file. But you can if you want, it's not going to break it, but you will have to sit there for a second. Has anybody already catted the file? Behind me, is it still running?
It was.
It was, right?
Beforehand.
Alright, so it's always important when if you're going to cat a file, it does what? Catting the file does what?
Opens the whole file.
Opens the file and prints it to the screen, right? So if you don't know how many lines or you don't know how big the file is and you catted the screen, it's going to just start printing all that there, so you're going to have to close the screen out or kill that part. And how, if it's doing it, if it's just printing, what can you do to stop it? I heard it over here. Control C, right? So if you do anything that you want to kill that part of it, just Control C and it will stop what it's doing, right? You can cat it if you just want to see. But another way to know what is in there, we know it's the number of markers. So the question is, how many markers are on the array that was used? Well, you can just look at the number of lines, right? Because we said each marker gets its own line. So how many markers are in that file? Exactly, 52,440, right? Right? So 41 lines, because one's a header. Very good. And again, if you brought that file over to Excel, you can just open it in Excel, use the way that you guys have learned now to put in nice little columns, see the number of lines, and know that something is there as well. Of course, that's not always possible if it's a giant file. Horse has an array that's over 670,000 markers. I think human has quite a decent one as well. And there's also a 2 million array, at least in horse for research. So you wouldn't be catting any of those files to screen because it would take quite a long time. So, what is the highest number of homozygous individuals at any marker in the array? So, what is the highest number of homozygous individuals at any marker in the array? So all it's asking is for any of the individual variants, which one has the highest percentage of homozygous individuals? Well, which column are you interested in looking at? And if you want a little bit of a hint, there's on the screen.
When you say markers, what do you mean?
SNPs. SNPs or loci. So it's a marker in the genome. So sorry if I use it interchangeably. A marker, no. So just remember, we're dealing with an array, and every array has a certain number of SNPs on it, right? Markers. So loci, marker, SNP just be used interchangeably in this context.
So you said something SNPs and then something else.
Marker, loci, like the location, loci. Yeah, L-O-C-I, loci. Okay. So I'm just using them interchangeably. But then, because in this context, it's the same. The location, the marker, the allele, they're all. In the context of an array, I would. For the most part. And for this test, it's fine. You wouldn't be penalized as long as it's in the right context. Because essentially, if you have 20 loci on the array, you have 20 markers. You're looking at 20 steps.
But there's 1, 2, 3, 4, 5, and 4 plus 5.
Yes.
Yes. So, what do you think is?
So, if you need to add together the homozygous cases and controls in your data, right? Because we said that there's no cases, there are no controls, it's all of them, right? So, but if you use this command, you can actually use the command. So it's a line trick to add the last two numbers together and print it out to a different file. That's what this command is doing. And it's printing out to the file name that you see there at the end, file name, summary combined.
Nice.
Once you do this, you can then sort that file on the last column. So now column number 6, right? And just show the largest ones. So you can sort it based on column 6. And just so, if you tail that file, the last value on the last line is the largest number of homozygous individuals of the 85 individuals in the analysis. And I would ask you, which chromosome is this location on? Sorry? 16, correct. So on chromosome 16. Who else got that? Still working on chromosome 16? Okay. So behind me, 16. No, not there yet. The wall, good. Thumbs up. Yes. Okay, so everybody got chromosome 16? Yes, chromosome 16? Okay, what can you now do with this information? What can you do? You've discovered that one location has quite a lot of homozygous individuals. How many individuals does it have? 6, so not that much, right? Because we said the population is not particularly inbred. But let's say it was 90% of the population had this. What can you now do? Some of you guys are going to say, well, yeah, you can literally go to the genome browser for that species, put it in this region, and see what you get. Right? And it could be something that's known, it could be something that's unknown, it depends on the population you're looking at. On a positive note, favorable note, it could be what we said before. It could be something selection, something that has been selected for. What could it be if it was more like, oh man, this is a small population, this is a concern? What could the gene it's tied to? So you find the gene and that gene's linked to what? That would give you concern or pause. It's probably a bit easier for some of the people who are in more animal livestock side. It could be disease, but the likelihood that it's a disease thing and what is quote unquote a healthy population is low. Because if it's something too severe, right, they would just die off, natural selection. But what could it be an early sign of a problem that's on the horizon? What do we know is tied with inbreeding? I might have heard it behind me, but homozygosity, okay? So homozygosity usually results from the mating of what? Closely related individuals, right? And one of the first things that we'll see from the animal side is as inbreeding goes up, so homozygosity goes up, the first sign of that usually tends to be fertility falls off a cliff, right? So fertility tends to drop. Dairy cattle's had this problem for a very long time, right? As inbreeding goes up, because they're selecting so heavily for milk production, fertility tends to drop quite rapidly. So usually one of the first indications of, oh, there might be some excess inbreeding is going to be fertility problem. Now the question we often get is, well, how much inbreeding is too much? And my response is always, I'll let you know once you pass it. Right? Because it's very hard to tell them where the cliff is, because it's going to vary by breed, it's going to vary by species, and it often varies by the very specific trait that you're looking at. But it's a real issue in livestock. As inbreeding goes up, performance suffers a little bit, but fertility takes the biggest hit. And it's not just a lethal variance, it's also like they failed to conceive. Because think about it, I think I mentioned earlier in this class that everybody has probably at least 100, in this room right now, everyone probably has 100 lethal variants in their genome, right? That's very, very rare, it's very uncommon. There's probably just random ones across, but if you constantly kept, if you went full on House of Dragons and you started marrying your siblings, right, over the generations, you would literally increase the frequency of those alleles, right? And if they're associated with anything that's embryonic lethal, you're going to see fertility take a hit. Any questions about that? No. Okay. So something that I think is very pertinent today, since everybody seems to be a bit stressed. So better to attempt something and do it great, to do something great and fail than to attempt to do nothing and succeed. I think that applies greatly to genomic stuff, right? Better to try something and it not work than just not try anything at all and hope that someone else does it for you. All right, so I encourage you guys to keep up with the fishing mentality, keep trying, keep pushing through. Do not be afraid to try things on your own. Like I said, all the tools are there, the pieces are there. Just because it's not an exercise that we've written doesn't mean that you can't come back and do it again. Now, with that in mind, there's an hour and a half left in class, right? For the next hour and a half, there are 2 questions that are coming up on the slides that I want you to work on in groups. The first group to give us the right answer does get a small prize, right? It is a very small prize, so it's not quite the level of a big gift card for coffee, right? I would also say, if you don't want to work on that, you can. I would encourage it because it will help practice skills. You do have assessments coming up. It is also a good opportunity if once you've solved those problems, instead of just leaving, you can stick around. We can answer some questions. We can help go through something that you're unclear on. And it's a great opportunity, if you haven't already done it, to make sure that you have put yourself in your groups for the group project and that you have all signed up and registered on the Canvas page. Because yes, the presentations are not until week 13, but you definitely don't want to put it off to the very end.
Did he
So I will put the first question
like
up
at the
on the
beginning
next
of
slide.
this? Sorry.
Once, if you're
Did
working
he
in a
assign
group
us
or
to
by
do
yourself and you get
in
the
this
answer and you
afternoon
just want to
session
tell us
or
what it
not
is,
yet?
we'll come
Not
by.
yet.
If it's right, you get a little prize.
Apparently it's just like, it doesn't have
We tried to copy the course and said, they may be in yeah, so if you go to it, you go full stop. Already is my it's already I just moved it to the subfolder. I was gonna put it on the yeah.
So if you change the directory into the group directory,
In group, well, so you have to go back into
Oh, okay.
so you just go into home, and into GEGE. And in the group
Oh wait, it was right there. Okay.
No, you actually aren't allowed to use the directory. I'm prepping Wait, what's the way you said Wait, if we both got that from the stuff on my if you do that and you're like, oh, yes, because it's alright. But I know nothing about horses.
All of them have the same number of
I don't think I'm gonna hate it because it's really interesting. I just think I don't need to know that ponies are horses. It feels like shorts.
Yeah. So I was going through the last parts of the activity, and basically we were supposed to get chromosome 16 for the, I think that's it. I got chromosome 9 initially, and then I looked through.
So what are you asking? So what are you
Oh,
asking?
it's just like, is this was correct?
What's correct? What are you saying?
The values for the.
So it's asking you what? Fortunately, you already did a pony. The pony is individual, specific individual.
Yeah.
Is the pony gray? How do you know? Is it gray?
Yeah, because you missed the 14 point here.
Okay. So basically, what the command is doing is
So what the command is doing, it's taking this file, your file, and it's printing its first column, second column, third column, fourth column, fifth column, and then the sixth column where the fourth and fifth column is combined.
Oh, okay.
You didn't put the fourth and fifth column, so it didn't read that.
Okay.
So total is 0.
Alright, thank you so much.
And that And how did you find that?
So then, because like there's a
I multiplied it by 4 and then added 6 because
And that just makes it
so what would be the swinger type of machine? Or it would be like yeah, so would the genotype be what? Well, I guess okay, that would make it I'm assuming that it's not the right location. Let's say, is this the best way to do it? That's great. So it's not like it's just another 125. So yeah, so you did it like a very forceful. Right. So how was I supposed to? So I'm sorry, I'm going to lead you for that. See if I can figure it out. But when you like, we can bring the map file. So you're doing the map file and do this. So you understood what the formatting is. This is great. Clean can also say well done. So we already have one person who's got it. You do get a prize. Since we're all about fishing, I figured worms were appropriate. We're not quite catching fish yet, but we're getting there. All right, sorry. So someone's already found the answer to the great question, but you can keep working on it if you like. That's fine. I'm not going to say the answer. And some person has already found it, so the prize is no longer available. All right. But what I'll do is I'll give you guys another 30 minutes or so. If people start figuring it out, then I'll switch to the next question. All right?
Not yet,
But it is in the notes, so if you do want to move to the next question now, it is in the notes. I just won't put it on the screen. Could be like, no, it could also be like, yeah, that's what it's, yeah. You know what the family life requires certain but also there's, after the right school, you'll start putting out some information, eventually you'll get to where you can actually use it, and that is figure out where or random individuals can do, whether it be humans, like on a European descent or an African descent, you can plot a genome database, genetically this is where you fit, and it's there if you breathe. So like certain unknowns, like what breed is this? The mixture is half it's very advanced. They have clones. And we don't know if they're doing a certain number of fields. Yeah, so that's what they do. It's pretty easy to do. Especially if you've got if it's a profile population, so horses, sorry, the example I would do is you might have a horse that's Japanese. So it will say like, oh, it's Mongolian. Because there's a lot of that horse. So he'll put in the reason. It's good when we have it now. In different places, we can put you in there. Oh no, you're not just from Germany, this town, this area, because that's where the family got it out. And there's one person in that area. That's why it's always a big bag. But yeah,
Just like, I don't have the